Hey, Hammy here, back with a second video from Chapter 7 on photosynthesis. In this chapter, we're going to look at plants as solar energy capturers, or the first step of photosynthesis. In order to capture solar energy, plants need something to catch the light. Okay, catching the light is going to be the role of pigments and also things we call photosystems, and we'll look at that in a minute. Pigments are chemicals that absorb certain wavelengths of light, okay, based on their color. The wavelengths that are not absorbed are reflected or transmitted, and so they look that color. So they absorb all the other wavelengths, but they reflect that back to our eyes, and that's what we see. If we look at different pigments found in chlorophyll, we see, and we run an absorption spectra on it, we can see what colors are absorbed and what colors are reflected. And so that graph kind of looks like a rainbow with a line on it, and I'll show you here on the next slide. And we'll see why chlorophyll looks green and why carotenoids, xanthophylls, some of the other things we'll see in our plant pigment lab, when we separate out those pigments, we'll see those different colors. Okay, so here are three things that you should kind of be familiar with. The electromagnetic spectrum, which you have probably done in previous years. This shows kind of increasing wavelengths or decreasing energy. Gamma rays being over on this end of the electromagnetic spectrum, radio waves over here. Okay, the portion that we call visible light is this skinny portion right here. And, you know, if you use a prism and you diffract that light, you bend that light out based on wavelengths, the different colors of light have different wavelengths. So if we use those frequencies, about 380 to 750, and we do absorption spectra of different pigments, you'll see chlorophyll A, chlorophyll B, right here, these lines. You'll see that they have peaks, okay, where they're absorbing a lot of light, right here in the blue-purple range, and also over here in the kind of the orange, reddish, and a little bit of yellow range. Notice what colors they are not absorbing right here. Yeah, green. So what color does chlorophyll look? Green. It's absorbing the other wavelengths, but reflecting green back to our eyes. So that's why chlorophyll is a green pigment, because that's what it's not using, it's reflecting. Also, same with carotenoids. You can see it absorbs a lot of the blues and blue-greens, but it reflects the oranges and the yellows. So it has more of an orange color because it's not absorbing it, it's reflecting it back to our eyes. If we take a plant and we shine different lights on that, different color bands of light, and we measure, this is called an action spectrum, not absorption, action. So how much, and often rate of photosynthesis can be like the amount of O2 bubbles being given off by a plant when you shine different wavelengths of light on it. You'll notice that here in the violets and blues, okay, you get a lot of photosynthesis. In the reds and the oranges, a lot of photosynthesis. Here in the green yellows, not much photosynthesis. So the action spectra very much reflects the absorption spectra because obviously the pigments are absorbing the light in those different wavelengths. Okay, now we want to get into the actual pathways. Okay, so the first one we call the light reactions, or oftentimes you'll hear it as the light dependent reactions, okay, because they are depending on light. You have to have light for this step. There is a non-cyclic and a cyclic pathway, and I'll demonstrate that here in a second. The goal of these light reactions is to capture the light within two photosystems. They have a pigment complex with like a little antenna, okay, which is the chlorophyll A. And this is all happening in those little stacks of pancakes or thylakoids in the thylakoid membrane. They're both cyclic and non-cyclic, produce ATP. Only the non-cyclic pathway catches electrons and puts them into NADPH. First is the non-cyclic pathway. Again, takes place in the thylakoid membrane. It uses two photosystems. PS just stands for photosystem 1 and PS2, photosystem 2. This one actually occurs first. Okay. We found it second. So they found this one first, so they named it PS1. But in the actual steps of photosynthesis, PS2 happens first. The non-cyclic pathway begins with photosystem 2. It causes an electron to be ejected from the reaction center. Okay, so you'll have a bunch of pigments in the membrane down here that are going to collect the light energy. And they're going to take that light energy, okay, the light that's coming in right here. They're going to take the light that's coming in. It's going to catch the light. It's going to take water, H2O, and it's going to split it. So it's going to make the electrons jump off of here. You're also going to get rid of two H pluses and a 1/2 O2. Okay. In other words, an oxygen atom doesn't just stay by itself. It'll quickly join to another water that's being split. Okay, that electron jumps up to electron carrier that's going to grab that, and it's going to pass it down an electron transport chain, and I'll illustrate that here in a second. And when it goes down that electron transport chain, it's going to pump hydrogens into the thylakoid space, and that's going to create a chemiosmotic gradient, which, if you remember from Bio 1, okay, it's going to, the hydrogens can escape through the ATP synthase, which is going to cause it to spin, which is going to turn ADP into ATP. And you'll see that here in a second. And then the electron down here we say is spent. And then that electron will be sort of re-excited back up here in photosystem 1, where it'll be picked up by NAD, if I can write here, NADPH, and be taken to the second step of photosynthesis. Okay, again, this is just the overview. Okay, we're talking about the first step of photosynthesis happening over here in the thylakoids. So you're taking in light, water. Okay, you're using light to split the water. You're giving off electrons to NADPH, also creating that chemiosmotic phosphorylation, or making ATP. And then also oxygen is going to be being given off as the byproduct. This is the more in-depth view. Okay, so we're starting over here. Here's that group of pigments that I talked about, and your chlorophyll A as your reaction center over here. Chlorophyll A. So in comes light. Okay, sunlight's going to come in here, zap it. Here's our water being split into hydrogen and oxygen. Here's our charged electron. And think of it, this as increasing energy level. So we say that electron goes up in energy to an electron acceptor. And then you have these little carriers here where the electron's going to jump from, it's going to be passed from carrier to carrier. And when it's doing that, it's going to pump in hydrogen ions into the thylakoid space. And these things are going to build up in here. Okay, and then down here, okay, is going to be down here will be the ATP synthase, okay, which is how they escape. Okay, when they escape, that's going to cause that to turn, and it's going to phosphorylate ADP plus an inorganic phosphate and make ATP. Okay, ATP, which is what they're showing here. Okay, once the electron is spent, once the electron gets down to here, in comes more light. It's re-excited to a different electron acceptor, and then it's passed on to NADPH plus hydrogen to make the electron carrier NADPH. Okay, this over here, this is photosystem 2. Remember, I said happens first. Photosystem 1 happens second. So this is non-cyclic. You can follow the flow of the electron through this whole thing over here, eventually to NADPH. This is taken along with the ATP to the second step of photosynthesis, to the Calvin cycle. Again, just some review here. Photosystem 2, again, has a pigment complex and electron acceptors. Receives the electrons from the splitting of water. Okay, and oxygen, this is where our product of photosynthesis, oxygen, is released. It combines with another oxygen to make O2 gas, which can be released from the plant. Okay, when that electron is excited, it goes to the electron transport chain, which has cytochrome complexes, plastoquinone. That's those little things where the electron's jumping, bump, 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 bump. Okay, it has a ATP synthase. It connects photosystem 2 to photosystem 1, and pumping that hydrogen into the stroma. From the stroma into that thylakoid space creates a chemiosmotic gradient, which ATP synthase is then going to be the opening for hydrogen ions to get back out. And this drives ATP synthase, turns it to turn this into ATP. And then the electron is picked up by photosystem 1, which has another pigment complex, which jumps it in energy. It's going to be picked up by an electron acceptor and added to NADP plus to make NADPH. So just kind of an overview in words here. I really like this picture here because this is showing the thylakoid here. Okay, this is showing a thylakoid, and remember, these are phospholipids. Okay, phospholipid bilayer, and here's photosystem 2. There's our antenna complex. Here's coming light. Light comes coming in, exciting the electron. Here's our water being split. Okay, so here's our electron being passed along these cytochrome molecules right here. Okay, and the plastoquinone. Okay, plastoquinone right there is going to, as the electron is shuttled along the electron transport chain here, here's our hydrogen ion that's going to be pumped into the space in here. Okay, now notice all the hydrogen ions in here. They don't like it in here. Okay, the spent electron down here can be used. Here's NADP reductase. Okay, so here's photosystem 1 right here. In comes more light. It's re-excited, and then that's used to reduce NADP plus into NADPH, which is going to go to the Calvin cycle. And those electrons are going to be used to reduce carbon dioxide, CO2, into our sugar. All these hydrogens can escape through our ATP synthase, which is going to cause it to turn ADP and an inorganic phosphate into ATP, okay, which is chemiosmosis or chemiosmotic phosphorylation. And that ATP, along with the NADPH, will also head to the Calvin cycle as well. Okay, so this is the first step of photosynthesis, the solar energy harnessing step. Okay, we're taking solar energy, putting that energy into the electrons from splitting water, and eventually either making some ATP through chemiosmotic phosphorylation or putting those high energy electrons from photosystem 1 into NADPH. So we have energy and high energy electrons that are going to go to the Calvin cycle to make our sugar. Again, a review, kind of in words, some overviews. The thylakoid space is the reservoir where those hydrogen ions build up. So every time water is split and the electrons are taken from it, every time it's oxidized, the two hydrogens that remain are also in that thylakoid space. Okay, and the transfer of electrons from the ETC is used to pump even more hydrogens into that space. Okay, and then the flow of hydrogen ions back through the ATP synthase causes it to spin and causes an enzymatic reaction to produce ATP. And like I mentioned many times, this is called chemiosmosis, or oftentimes I refer to it as chemiosmotic phosphorylation, because we are phosphorylating or adding a phosphate to adenosine diphosphate to make adenosine triphosphate. There's kind of a special focus in your book talking about climate change and also rainforest destruction. It's important to understand that plants, as autotrophs, as photosynthesizers, are taking CO2 up out of the atmosphere. So a lot of us know that burning fossil fuels, okay, burning old organic matter puts CO2 into the air. So it's important to have trees because trees take the CO2 out of the air and lock them back into sugars and other organic compounds. Okay, so we call this we call trees and plants, especially trees, because they have such biomass, we call them a CO2 sink. So deforestation is besides burning fossil fuels, deforestation is really causing a lot of that increase in atmospheric CO2. And that's where this problem becomes, because throughout history, 1880 to 2010, when we have, as long as we've had records of this, you see that as we've increased CO2 levels, you look at the degrees in Fahrenheit or Celsius, we've gone up just over an average mean global temperature of about 1 degree Celsius. So the question now becomes, what is going to be the result of this? Will it drastically change things? Will it change weather patterns? Will it change how we grow crops, where we can live, the ocean life? All that stuff are big question marks about. They're looking at how will this affect it. And how we deal with how many trees we have on this planet really much affects that CO2, which goes back to what we're talking about in the chapter here, photosynthesis. Anyway, that's the first step of photosynthesis. Hammy, out.